We observe in table 14.1 that there were different values for the melting points of fatty acids. If we pay close attention to the data that is on the right side of the slides, we are going to observe that here we have examples of two different fatty acids. We have on the left side stearic acid, which is a saturated fatty acid. And then on the right, we have oleic acid, which is a monounsaturated fatty acid. Both of these fatty acids have 18 carbons in length. But if we pay close attention to the data that it is provided, we can observe that stearic acid has a melting point of 69 degrees Celsius, while oleic acid has a melting point of 14 degrees Celsius. So even though these molecules have the same number of carbons, we can observe that there's drastic changes or drastic values, if we put it in that sense, in their melting points. Let's explain why that is. Let's remember that fatty acids, like many lipids, are non-polar molecules. So the attractive forces that are going to be present in these molecules are going to be London dispersion forces. Let's also point out that even though the molecule has a polar side, because the amount of non-polar character or the non-polar bonds that exist in the molecule, overall, the molecule is considered non-polar. And that is true also of the monounsaturated fatty acids. Now, even though London dispersion forces are present in these molecules, one of the things that happens within these structures is the ability for these London dispersion forces to actually interact with one another. The observations that we see when it comes to the differences in the melting points between a saturated fatty acid and an unsaturated fatty acid has to do with the packing of these molecules with each other within the substance. It is known that saturated fatty acids are going to have the ability to pack together because they are going to be able to be stacked. So if we want to use an analogy in order to understand the stackability of fatty acids, we can visualize that saturated fatty acids are going to be able to be stacked like Pringle potato chips because of their shape, because of the bonds present in stearic acid. We can actually pack these molecules one on top of each other. In the case of unsaturated fatty acid, we see that these molecules, since it has a carbon-carbon double bond, and you can see the cis configuration of it, are not going to have the packing ability that saturated fatty acids have. What happens is that the carbon-carbon double bond introduces to the molecule what is called a kink. And these kinks are going to basically prevent them from packing together as closely as the chains of saturated fatty acids. As an effect, we see that the melting point of unsaturated fatty acids is decreased. Now, if we utilize the same potato chip analogy to look at the packing ability of molecules, unsaturated fatty acids are going to be more like Lay's potato chips or kettle potato chips, which because of the shape and the kinks that are in them, they will be able to pack, but maybe not as well as the saturated fatty acids. Let's now continue by looking specifically at saponifiable simple lipids. And at first, we are going to discuss the structure of waxes. As I mentioned previously, simple saponifiable lipids are going to have an ester bond. And in the case of a wax is composed of a long chain fatty acid along with a long chain alcohol. As an example of a wax, here we have the structure of beeswax, and we can see the long chain carboxylic acid portion along with the long chain alcohol portion. Waxes often occur in nature, and they are going to be part of protective coatings that are present in feathers, in fur, in skin, leaves, and even fruits. Let's understand that waxes can be present in plants and they can also be present in animals. An example of a wax is going to be sebum, is something that it is secreted from the glands of the skin and it contains different waxes. We can also see in figure 14.5, specifically beeswax. And beeswax has also been utilized in the cosmetic industry for lip balms. Beeswax additionally has been used to make candles, hand creams, moisturizers, and even eyeshadow. The main thing and the main role of waxes is that it's going to be able to keep skin soft and prevent dehydration. Now let's look at another simple saponifiable lipid. Now let's look at triglycerides. Triglycerides are also known as triacylglycerols. These molecules are triesters of glycerol in which all three alcohol groups are sterified. So when we look closely at how do triglycerides form or what are the components of triglycerides, we have one molecule that has one, two, three hydroxyl groups that is going to be acting as our alcohol. And we know that molecule as glycerol. Hmm. Glycerol can bind to one, two, three fatty acids. And when it comes to this, via the reaction of esterification, we can see how the alcohol group and the carboxylic acid group within the fatty acid can sterify to make a triacylglycerol. So in equation 14.1, we can see the formation of a triacylglycerol from glycerol and three molecules of stearic acid. Now, it is important to determine that when it comes to fatty acids can have the same, or I should say, the triglycerides can have the same fatty acids in all three positions, or sometimes they can be mixed. We're going to see mixed fatty acids a little bit later in the presentation. When it comes to triacylglycerols, some of them we categorize them as fat, and others we categorize them as oils. The basic difference between a fat and an oil is that fats are going to be a triglyceride that is solid at room temperature. And usually it comes from animal sources like meat, whole milk, butter, or cheese. Oils, in the other hand, are going to be triglycerides that are liquid at room temperature, and they are obtained from plant sources like palm oil or coconut oil. When it comes to looking at the composition between fats and oils, looking specifically at figure 14.10, we can see a comparison of saturated and unsaturated fatty acids that are present in some foods. We can see that in these bar graphs, we have on the left animal fats, and on the right, we have vegetable oils. Animal fats are mostly triglycerides that have saturated fatty acids. And as we saw before, if you have a high number of saturated fatty acids, you tend to have a higher melting point. Now, in this graph, what we are also seeing is that when it comes to the percentage, meaning the blue area of saturated fatty acids that are present in these molecules, we see that there is large areas of blue in many of the things that are animal based and they have fat in it. On the other hand, when we look at vegetable oils like corn oil, like soybean oil, we can see that the area that is in larger amount is going to be the pink area, which is going to be representing the unsaturated fatty acids that are in those triglycerides. It is important to note that when it comes to vegetable oils, they're going to have a lower melting point, but in a way, it is because of the number of unsaturated fatty acids that we have in those substances.